Introduce today's speaker, Emily Greengard, who is an assistant professor here in the Department of Pediatrics at the University of Minnesota. Originally from St. Louis, Missouri, she went and finished an undergraduate degree at the University of Wisconsin-Madison, majoring in psychology. Then went on to medical school at St. Louis University School of Medicine, was a resident at Northwestern University, and then did fellowship training at Children's Hospital of Philadelphia before coming here to Minnesota. So her career is really, she's focused on translational clinical research in pediatric oncology with a specialty in neuroblastoma and sarcoma. She was named a rising star in Minneapolis St. Paul magazine among doctors for many years, and now she's a top doc. So she's risen through the ranks of that magazine. She's been given the Innovator Award in Translational Research in the Department of Pediatrics. She's highly active in running clinical trials here at the University of Minnesota. There was pages and pages of them. I didn't want to print them all out. Including work that's close to stuff that we've done in my own lab, because she's been able to lead a clinical trial targeting SEMA4D for osteosarcoma, which is something that came out of work that Brandon Moriarty did while he was working with me and in his own lab, along with Brandon Speester, who's back there. But today she's going to tell us about targeting ALK in neuroblastoma as a paradigm for targeting molecular drivers in pediatric cancer. So thank you.
Thank you, David, very much for the introduction, and to the Cancer Center for asking me to speak today. And I chose to talk about some of the work I've done in translating the amazing work that my colleagues have done in the lab looking at ALK inhibition in neuroblastoma, translating that from the lab to the patient, and thinking of this as a paradigm for targeting molecular drivers in pediatric cancer. And I chose not to talk about SEMA4D because people smarter than me in the audience, like David Largaspada and Brandon Speester, and maybe Brandon Moriarty, might have been here. So I just wanted to say that I have no financial disclosures. I will discuss the off-label use of Crizotinib and Lorlatinib in neuroblastoma. I just wanted to start a little bit with the rationale for developing new drugs for pediatric cancer, which is probably quite obvious to a lot of people in this audience, why we need new drugs. However, we do know that the 5-year survival for children with pediatric cancers now is greater than 80%, which is excellent. And we've had significant improvement in survival over the past 30 years, with survival rates less than 60% in the 1970s, and now greater than 80%. Although, not all pediatric cancers are created equal, and there are still some types of pediatric cancers in which survival is not that good. In addition, children who are treated for pediatric cancers have acute and late effects of their treatment that cause many challenges for these patients. And these late effects include growth and development issues, organ dysfunction, difficulty with infertility, secondary cancers, and relapse of their primary cancer, as well as a multitude of psychosocial and mental health issues. There are a lot of challenges in drug development for childhood cancer. And a lot of this stems from the fact that pediatric cancers are different than adult cancers. They differ in the tissue of origin, in the pathogenesis and genetic alterations that drive the cancers, or the lack thereof, in their growth characteristics and their sensitivity to drugs. In addition, childhood cancer is rare, and our initial multimodal treatment oftentimes is curative, which is wonderful, but leaves us with a limited number of patients to study in early phase clinical trials. In addition, we're typically studying children that have relapsed or refractory disease and are heavily pretreated, and therefore might have greater toxicities and a decreased response as compared to children who might be treated with novel agents up front prior to being treated. Neuroblastoma, which is the most common extracranial solid tumor in children, is an example of a disease in which we still need novel therapies to improve outcomes for these children. This over here on the left is a breakdown of the incidence of cancer in children 0 to 14 years of age. You can see that neuroblastoma accounts for 7% of pediatric cancers. It's quite less common in older children and adults. Yet, despite only accounting for 7% of pediatric cancers, it accounts for 15% of childhood cancer deaths. Neuroblastoma is really a paradigm for the prognostic utility of both biologic and clinical data in predicting outcome and risk for disease recurrence and death. And this chart is not for you to read every little detail, but just to point out that neuroblastoma risk assignment, so the risk of recurrence there are several factors that go into this. This includes the stage of the disease, the age of the patient, the histologic category of the tumor, the grade of the tumor differentiation, and then several genetic alterations within the tumor, including the presence or absence of MYC amplification, segmental chromosomal aberrations, as well as whether the tumor itself is a diploid tumor or hyperdiploid tumor. And outcomes for children are highly dependent on risk stratification. Children stratified as having low-risk disease, nearly all survive, whereas children with high-risk disease, the large majority of them succumb to their disease. Now, fortunately, this Kaplan-Meier curve that shows event-free survival by risk stratification is a little bit outdated, and survival for children with high-risk disease is better now in 2020 than it used to be, but it's not perfect, and it requires intense multimodal therapy to cure children with high-risk neuroblastoma of their disease. So the treatment for neuroblastoma is quite long. It takes about 18 months. It includes an induction phase, which includes 5 cycles of chemotherapy, as well as surgical resection of their primary tumor. And then a consolidation phase, which includes 2 rounds of high-dose myeloablative chemotherapy, followed by stem cell rescue, and local radiation therapy to their primary tumor. And then children go on to receive post-consolidation therapy, which is 6 months of immunotherapy. With this therapy, we have had significant improvement in survival, and the most recent clinical trial that was executed by the Children's Oncology Group randomized children with high-risk neuroblastoma to receive either the standard of care, which was a single round of myeloablative chemotherapy followed by stem cell rescue, versus tandem transplant, or 2 rounds of myeloablative chemotherapy followed by stem cell rescue. And the Kaplan-Meier curve on the left shows event-free survival for children who were randomized to a single transplant versus tandem transplant, and then went on to receive immunotherapy. And on the right is overall survival. And you can see that children randomized to receive tandem transplant in the yellow line did statistically significantly better than children randomized to single transplant, with a 3-year event-free survival of about 76%. So a dramatic improvement from where we were 10, 20 years ago for high-risk neuroblastoma. But these children have a multitude of late effects. So this graph over here is looking at the mean number of follow-up years after treatment, and on the x-axis and on the y-axis is looking at the number of late effects. And you can see late effects increases over time, and the late effects are quite variable and affect multiple organ systems. So on the right here is looking at, on the x-axis, onset of late effects in years, on the y-axis, the various late effects, and you can see things like hearing loss, renal effects, neuropsychological effects occur early on, and then we run through the gamut, and cardiac late effects and eye late effects occur later on. So given both the significant toxicities of therapy for high-risk neuroblastoma, as well as still the suboptimal survival. Really, novel therapies are needed for children with high-risk neuroblastoma. And one of the novel therapies that I have been really interested in and have focused the first 10 years of my career on is anaplastic lymphoma kinase, or ALK, as a potential therapeutic target in neuroblastoma. So ALK was first described in anaplastic large cell lymphoma, hence the name, and it was described as a chromosomal translocation that resulted in a fusion protein with the ALK intracellular region fused to the nucleophosmin N terminus. This leads to constitutive activation of the ALK kinase domain, and similar fusion transcripts are seen in non-small cell lung cancer. So here, this down here just depicts the translocation that leads to constitutive activation of the tyrosine kinase, and then downstream activation of several pathways, including ERK1, ERK2, the mTOR pathway, and essentially cell cycle progression. ALK over the past many years has really been found to be an oncogenic driver across several malignancies, both hematologic and non-hematologic malignancies. And I'll talk a lot more about the role of ALK in neuroblastoma, but ALK has been found to have a role in, as I said, anaplastic large cell lymphoma, non-small cell lung cancer, as well as inflammatory myofibroblastic tumors. So it has been known for about 20 years that ALK is expressed in nearly all neuroblastoma tumors. So the first reports back in 2000 came out of a study that looked at 56 cancer cell lines, including 29 lines of neural origin. In addition, this group looked at 24 neuroblastoma primary tumor tissue specimens that were obtained from the Pediatric Oncology group, and found that there were found ALK transcripts and proteins in 45% of the cells of neural origin, cell lines of neural origin, but 64% of neuroblastoma cell lines and 92% of neuroblastoma primary tumors. The mRNA encoding ALK protein was not detectable in any normal tissue or neoplastic hematopoietic tissue tested, except for in ALCL. Following this, in 2008, a sort of landmark study found that germline ALK mutations result in a familial neuroblastoma syndrome, or a neuroblastic tumor predisposition syndrome. This was discovered when 20 families that had multiple cases of neuroblastoma were evaluated and had a genome-wide scan for linkage at 6,000 SNPs executed. And this revealed significant linkage signal at chromosome 2p in the 18 families that they were able to evaluate. 2 families were excluded due to insufficient DNA. And they found the predisposition locus to be defined at chromosome 2p23 to p24, which is where both MYC-N and ALK are located. But they did a resequencing of the MYC-N coding regions in the probands and found no disease causal sequence variations in MYC-N. So it was thought that ALK was the driver and the predisposition locus. So this is just, I don't expect you to see all the wording on this, but just an example of 8 of the pedigrees that they looked at in this study. And you can see the black boxes and circles, or squares and circles, depict individuals that are affected by neuroblastoma. So these families each had several individuals affected by neuroblastoma. And it was found that these families all had mutations in the ALK-tyrosine kinase domains located at R1192P, R1275Q, and G1128A. So while this work was going on that discovered these germline mutations, there were several investigators looking into somatically acquired ALK mutations in neuroblastoma, and certainly did find that activating ALK mutations are oncogenic drivers in neuroblastoma. So this here is the ALK gene, and when we focus on the tyrosine kinase domain, you can see we have the germline mutations I listed below, but also several mutations were found in neuroblastoma tumor tissue. that were not found to be germline mutations. Most of these mutations, somatic mutations, are located on 3 hotspots at F1174L, F1245, and R1275. And over here, this graph really shows what I sort of discussed before, but it's looking at ALK expression in mutated cell lines that show that ALK is highly expressed. In wild-type neuroblastoma, ALK is expressed, but not as highly as in mutated. And then there is low-level expression in fetal brain, although there is no expression of ALK in any other normal tissue within people. And down here, you can see that in mutated ALK cell lines, these first 3 cell lines, ALK is expressed uniformly and phosphorylated, whereas in wild-type neuroblastoma cell lines, ALK wild-type cell lines, ALK is expressed in some of the cell lines, but not all yet, but not phosphorylated. So further studies looking at ALK and, as an oncogenic driver in neuroblastoma were undertaken through the pediatric target study, most of this done at the Children's Hospital of Philadelphia. So in this study, they looked at 1,600 diagnostic neuroblastoma samples and found and did genomic, biochemical, and computational analysis for ALK mutations. They found that ALK-tyrosine kinase mutations occurred in 8% of the samples at the 3 hotspots I mentioned before, and also identified 3 minor sites. Biochemical and computational analysis studies were able to distinguish oncogenic or activating ALK mutations from non-oncogenic mutations. In addition, they found that 15% of cases demonstrated ALK copy number gain, but only 2% revealed high-level ALK amplification, and this was occurring exclusively in patients whose tumors also had MYC-N amplification. So to further investigate the role of ALK in neuroblastoma, studies were done looking at siRNA knockdown against ALK in comparison to a negative control. And this is depicted in several of these graphs over here, in which knockdown of ALK is the blue line and the red line is the negative control. And up here, you can see in an ALK mutated cell line, knockdown inhibits tumor cell proliferation and growth. This is the ALK amplified cell line. You can see the same thing. And then these down here are all ALK wild type cell lines, where knockdown did have an effect in one cell line, but not in the other 2 studied. And the graph over, this is just a graphic representation over here of these, a bar graph representation. And down here, this cell line, Kelly, is an ALK mutated cell line. And the cell line below is an ALK wild type cell line. And you can see that while ALK is knocked down at various time points in both cell lines, when we flip it over to the impact, cell growth is only inhibited in the ALK mutated cell line and not the ALK wild type cell line. So again, this is just a blow up of the tyrosine kinase domain of the full-length ALK, showing the 3 hotspots that we know occur in, are the most common in ALK, somatic mutated ALK. So really importantly, ALK status negatively impacts prognosis in neuroblastoma. So these 2 Kaplan-Meier curves on the left are event-free survival, are looking at event-free survival. On the right are looking at overall survival. The top panel are all patients with neuroblastoma, regardless of risk, and the bottom panels are patients with high-risk neuroblastoma. And what you can see is that patients whose tumors do not harbor an ALK mutation are in blue. Those whose tumors harbor an ALK mutation, or amplification, I should say, are in red. And you can see there's a statistically significant difference in survival, with worse survival for children with ALK mutated or amplified tumors. So because of the role of ALK as an oncogenic driver in neuroblastoma, clearly ALK became a target of interest for treatment of kids with ALK-aberrant neuroblastoma. So the first studies were done at the Dana-Farber Cancer Institute looking into ALK as a therapeutic target when they used a high throughput platform to screen cancer cell lines. And they looked at 602 cell lines with, and treated them with various ALK inhibitors, including this one, which we now know as Crizotinib. They found growth suppression occurred in a subset of human cancer-derived cell lines known to harbor ALK gene rearrangements or amplification. And this was also during the time that the somatic ALK mutations in neuroblastoma were coming to light. But it was known that these cell lines had either rearrangements or amplification. And they saw efficacy in ALCL, non-small cell lung cancer, and neuroblastoma cell lines. So several other preclinical studies were done to evaluate Crizotinib, a first-generation ALK inhibitor's role in the treatment of high-risk neuroblastoma. So Crizotinib is an orally bioavailable small molecule inhibitor of the c-Met, HGFR, and ALK receptor tyrosine kinases. And as I said before, extensive preclinical data demonstrate potent inhibition in multiple different cancer cell lines and tumor regression, which I'll say a little bit more about in mouse xenografts. I just wanted to talk a little bit about the timeline for development of Crizotinib because it is quite interesting, I think. So the boxes in the purplish color are the development of Crizotinib in translocated ALK in non-small cell lung cancer, whereas the blue boxes are the development of Crizotinib in neuroblastoma. So, you know, ALK was discovered in ALCL in 1994. And then if we fast forward to about 2005, this is when the phase 1 trial of Crizotinib in adults took place, which determined the maximum tolerated dose to be 250 milligrams twice daily. If we move forward to 2008, this is when the identification of ALK as both germline and somatic mutations in neuroblastoma was discovered. And then in 2009, the first phase 1 trial of single agent Crizotinib in neuroblastoma began. So that's actually a pretty quick timeline for moving forward in children, but the entire timeline is quite long. And then in 2012, we opened a phase 1 trial of Crizotinib in combination with chemotherapy for children. So a little bit more about the preclinical studies of ALK in neuroblastoma. This is a dose response curve for 4 neuroblastoma cell lines looking at different exposure to different doses of Crizotinib. And the green line is ALK amplified neuroblastoma cell line, whereas the other three cell lines are all ALK mutated. And what you can see is that the red cell line with an R1275 mutation and the ALK amplified cell line are fairly sensitive to Crizotinib, whereas the other 2 cell lines are fairly resistant. However, this resistance can be overcome with higher doses of Crizotinib. So this helped us as we were moving forward with the early phase trials of Crizotinib and provide a rationale for really maximizing the recommended phase 2 dose and pushing the dose as high as we could, given that some ALK mutations are more difficult to inhibit relative to translocated or amplified ALK. So once the adult phase 1 trial of Crizotinib was complete and the recommended phase 2 dose was known, and we had sufficient preclinical data in pediatric tumors, phase 1/2 trial of Crizotinib opened through the Children's Oncology group and was chaired by Yael Moshe at the Children's Hospital of Philadelphia. This study used a rolling 6 phase 1 dose escalation design, and it enrolled patients at the 80% of the adult maximum tolerated dose as the starting dose for Crizotinib. And the endpoints of this study were really to understand the maximum tolerated dose, safety, tolerability, and pharmacokinetics of Crizotinib in children. So as you can see, this is a summary of the cycle 1 dose-limiting toxicities that help to define the maximum tolerated dose. So the dose, first dose level was 100 milligrams per meter squared per dose BID. and escalated all the way up to 365 milligrams per meter squared per dose BID, at which dose level 2 out of the 6 patients had dose-limiting toxicities, and this exceeded the maximum tolerated dose. So the maximum tolerated dose and recommended phase 2 dose was found to be 280 milligrams per meter squared per dose BID. 17 patients were evaluable at this dose level, and none of the patients had dose-limiting toxicities. Of note, this dose is significantly higher than the adult recommended phase 2 dose of a flat dose of 250 milligrams BID. Now, when we look at the responses for children enrolled on this trial, you can see that there were 9 patients with anaplastic large cell lymphoma enrolled, and 8 of those 9 patients had either a complete response or a partial response, and the 9th patient had stable disease. Results were also encouraging for inflammatory myofibroblastic tumor, but unfortunately not quite as encouraging for ALK-mutated neuroblastoma. And 1 of the 11 patients with known ALK mutations had a response. 3 patients had stable disease. Of course, there were 23 patients with neuroblastoma enrolled in which the ALK status was unknown, and 1 of those patients had a response, and 5 patients had stable disease. And this is just a depiction of a neuroblastoma patient with a germline ALK mutation who had a response. And this is their MIBG scan, which is a way we stage and follow neuroblastoma prior to starting Crizotinib. And then at the 7th cycle of Crizotinib, you can see a significant decrease in the disease burden. Now, you might wonder, with the lack of responses, yes? Yes. Most all anaplastic large cell lymphomas carry an ALK translocation, so it wasn't confirmed on the trial. I'm going to repeat the question, sorry. The question was for the anaplastic lymphoma kinase patients, was the ALK status known for those patients? And really, for anaplastic large cell lymphoma, most all patients have an ALK translocation. And so while the study didn't require confirmation, it's presumed that those patients all had ALK translocations. So, one might wonder why we would want to further develop Crizotinib in neuroblastoma, given the lack of a robust response in our phase 1 trial. So just to speak a little bit more about that, we really learned that ALK mutations certainly have differential sensitivity to Crizotinib. And 2 of the ALK driver mutations observed in neuroblastoma, the F1174 and 1196, have arisen as acquired Crizotinib-resistant mutations in tumors that are driven by oncogenic ALK fusion proteins, so particularly in non-small cell lung cancer. So primary resistance to Crizotinib really is a major concern for neuroblastoma. And studies were done to evaluate this differential sensitivity to Crizotinib. And the sensitivity can be predicted by the Km of ATP, in that a lower Km would predict a higher ATP affinity for these mutations. And since these inhibitors are ATP competitive inhibitors, having a higher affinity would make it less sensitive or more resistant to Crizotinib. And then also looking at the IC50s of Crizotinib in these various mutated cell lines, the response could be predicted. So to highlight a few, the F1174, which has a low Km and a high Crizotinib IC50, is predicted by both mechanisms to be resistant to Crizotinib, and indeed it is. Whereas the the L1196 mutation has a high Km, so it would be predicted to be sensitive to Crizotinib, but a really high IC50, and actually has been found to be resistant to Crizotinib. Whereas the R1275Q has a high Km, which so predicted to be sensitive to Crizotinib, as well as a lower IC50. So both ways predicted to be sensitive to Crizotinib, and indeed it is. So although there is Crizotinib resistance in some of these ALK-mutated cell lines in neuroblastoma and ALK-mutated tumors, resistance can be overcome by combining Crizotinib with conventional chemotherapy. So all 4 of these neuroblastoma xenografts that were studied have relative resistance to single-agent crizotinib. So you can see that these mice, these xenografts, were treated with single-agent crizotinib in red, topotecan and cyclophosphamide alone, so conventional chemotherapy alone in purple, and then a combination of crizotinib and chemotherapy in green. And while you can see this cell line is somewhat sensitive to single-agent crizotinib, these other cell lines are really quite resistant with very little reduction in their tumor volume with single-agent crizotinib. When combined, you can barely see these green lines at the bottom because tumor volume and growth is completely inhibited, except for in this last model in which it's inhibited for about 4 weeks and then it breaks through. So this data that suggests that we can overcome crizotinib resistance by combining it with chemotherapy led to rationale for our phase 1 trial of combining crizotinib with chemotherapy in children with relapsed and refractory solid tumors and anaplastic large cell lymphoma. And I had the honor of chairing this study. And this study was a bit of a challenge, so you'll hear a little bit about it. But it was executed using a 3+3 dose escalation schema for dose escalation of crizotinib with fixed dosing for our chemotherapy backbones. And because we're combining it with chemotherapy, we started at 60% of the recommended phase 2 dose for crizotinib that was defined from the prior trial of single-agent crizotinib, because we were worried about overlapping toxicities with the chemotherapy, particularly hematologic toxicity. And we used a crizotinib oral solution to start because most of our kids with neuroblastoma are quite little and cannot swallow pills. And we were really interested in looking at the maximum tolerated dose of crizotinib when combined with chemotherapy, as well as safety, tolerability, and pharmacokinetics. So we had 2 chemotherapy backbones that we used on this trial. Part A evaluated topotecan and cyclophosphamide once daily for 5 days. And the rationale for this is that this is a combination that's used in upfront treatment for neuroblastoma, as well as in a lot of sarcomas. In addition, Part B looked at vincristine and doxorubicin, as these drugs are used in neuroblastoma, but also in leukemias, lymphomas, including anaplastic large cell lymphoma. So we enrolled patients concurrently on these 2 parts of the trial with the 2 chemotherapy backbones. And on Part A, this is again looking at our cycle 1 dose-limiting toxicities. On Part A, with topotecan and cyclophosphamide, we escalated to 215 milligrams per meter squared per dose BID, and 1 of our 4 patients had dose-limiting toxicities of diarrhea and dehydration. On Part B, we were able to escalate to 280 milligrams per meter squared per dose BID, but both of our patients at that dose level had dose-limiting toxicities of nausea, QTc, prolongation, and dehydration. So really 215 milligrams per meter squared per dose BID was defined to be the maximum tolerated dose of this oral solution. But you can see that a lot of the toxicities were gastrointestinal toxicities. And we actually know that the oral solution of crizotinib is not palatable at all. Children hate it. And we did a taste questionnaire in this study that confirmed that. And we asked kids several questions. And overall, the kids really did not like crizotinib at all. They weren't really sure if it was easy to swallow or not. That's a hard question for kids, I think. But they did not like the flavor, the smell, the feel, or the overall taste. And so we were really quite concerned that the toxicities that we were seeing were more due to the poor palatability of the oral solution rather than true drug toxicity. So for that reason, we opened Part C of the trial to study formulated capsules, which was difficult because this limited our younger patients who are not able to, limited enrollment for our younger patients who are not able to take formulated capsules. Fortunately, Pfizer at the same time was developing a microsphere formulation, a novel formulation to try to address this issue. So on Part C of the trial, we escalated to 280 milligrams per meter squared per dose BID again, but 2 of our 3 patients had dose-limiting toxicity of ocular toxicity and hypotension. At the 215 milligram per meter squared per dose level, only 2 of our 8 patients had dose-limiting toxicities of esophageal pain and hematuria. So we completed that part of the study, And then we moved on to studying Crizotinib microsphere sprinkles, which were developed to really address the palatability issues of the oral solution. And they were developed to allow for dosing in children who are unable to swallow capsules or were too small to achieve accurate dosing with capsules, even if they were able to swallow them. And these microsphere sprinkles contain a pH trigger membrane. So they're insoluble in the mouth where you have a neutral pH, and then soluble in the acidic pH of the stomach. So you can mix these sprinkles with water, the kids can take them, and then the drug will not be released until it reaches the stomach. And this also helps to achieve a taste neutral profile. So we opened part D of our trial to use the microsphere combination, the microsphere in combination with topotecan and cyclophosphamide. But surprisingly and unfortunately, we ran into significant dose-limiting toxicities, even at the low dose of 165 milligrams per meter squared per dose BID, with 3 of our 5 patients, evaluable patients, having dose-limiting toxicities of thrombocytopenia in 3, neutropenia in 2, diarrhea, dehydration, and hypokalemia. So we actually aborted the trial at this point. And we know that Crizotinib doses less than 165 milligrams per meter squared will not inhibit ALK, even when combined with chemotherapy. And so there was not a good rationale to further develop these microsphere, the microsphere formulation. Interestingly, when we looked at the pharmacokinetics of the 3 different formulations, pharmacokinetics did not explain our toxicity. And you can see the blue circles are patients treated with the oral solution of Crizotinib. The red squares are the formulated capsules, and the green diamonds are the microspheres. And on the x-axis is the dose administered. On the y-axis is the Crizotinib area under the curve, or exposure. And you can see that the patients who receive the microspheres all fall. Their AUC is quite low compared to some of the patients who got oral solution or formulated capsules. So it still remains a little bit unclear as to why we encountered such a significant increase in dose-limiting toxicities in the cohort that received the microspheres. Nonetheless, we were able to determine that our maximum tolerated dose for both the oral solution and formulated capsules when combined with chemotherapy was 215 milligrams per meter squared per dose BID. And we had the data we needed to move forward with integrating Crizotinib into the upfront treatment for children with high-risk neuroblastoma. So ANBL 1531 is this trial. It opened in 2018. And this is a large trial for children with high-risk neuroblastoma, with one arm of the study being for kids whose tumors harbor an ALK aberration. And the primary objective of this arm is to determine whether the addition of Crizotinib to standard multimodal therapy for patients with high-risk neuroblastoma whose tumors harbor activating point mutations and or amplification of the ALK gene results in a superior 3-year event-free survival compared to contemporaneously treated cohort of patients whose tumors harbor or lack ALK aberrations. And because this is a known molecular driver for the subset of patients, we're actually looking for quite a big difference with a 20% improvement in event-free survival as compared to the other population. So we do have some secondary objectives in this trial, of course, that are to look at the toxicities and evaluate the relationship of response rate to event-free and overall survival and induction response rates. And then several exploratory aims to determine whether the efficacy of adding Crizotinib to our backbone is associated with specific ALK mutations or amplification, to characterize change in circulating tumor DNA, particularly ALK, throughout treatment, and then to compare the late toxicities of treatment for children treated with Crizotinib to those who aren't treated with Crizotinib. So the eligible ALK aberrations include the known ALK-activating mutations, as well as ALK amplification, which is defined as greater than 10 copies of the ALK gene. And here's our schema, which, as you can see, if you remember the schema I showed you early on for standard of care treatment for kids with high-risk neuroblastoma. For these children, all children with high-risk neuroblastoma can be enrolled on this trial, and then their tumor undergoes ALK testing during their first cycle of chemotherapy. If their tumor is found to have an ALK aberration, then they're assigned to Arm E of the trial, which is the integration of Crizotinib throughout all of their therapy, although it's held for short periods of time throughout. And then we add in a continuation phase in which they're treated with Crizotinib alone for about 12 months. So this definitely extends the length of their therapy. So this trial, as I said, is a large trial for all kids with high-risk neuroblastoma. And we anticipate for the entire trial 774 kids to be enrolled over 5 years. For the ALK-aberrant arm, our assumption is that 14% of children enrolled will have ALK aberrations, about 10% mutations, 2 to 4% amplification, and then will be non-randomly assigned to the arm in which they'll receive ALK-targeted therapy. So this would lead to about 108 patients over 5 years, or 22 patients per year. And one of the challenges that we've had thus far is accrual to this arm of the study. So the study activated 22 months ago, and we've enrolled about 221 patients as of last month, which is good, which is right on target. But only 7% of the children have been found to have tumors that harbor an ALK aberration. And most of these are mutations. Only 2 of the kids have been found to have tumors with ALK amplification. So we're well below our expected accrual of 22 patients per year. And we know that part of the challenge is getting the submission of adequate samples to do the ALK mutation analysis, as we've had about 10% unsatisfactory results. But there's more to it than just that, and we're trying to get to the bottom of it. So as we try to kind of optimize our approach to targeting ALK in children with neuroblastoma, we also are thinking about whether Crizotinib is truly the best drug to use, and the answer is that it probably isn't. So looking at next generation ALK inhibitors, Lorlatinib has really become of interest in a lot of ALK aberrant diseases, including neuroblastoma. So Lorlatinib is an orally bioavailable, potent, and selective inhibitor of ALK and ROS1. It's a macrocytic ATP competitive inhibitor. It has a low propensity for P-glycoprotein-mediated efflux, and it has enhanced blood-brain barrier penetration over Crizotinib, which is highly important in neuroblastoma, where the central nervous system can serve as a sanctuary site, and it is not uncommon for kids to have central nervous system relapses. So Lorlatinib has been studied in preclinical models of ALK-mutated neuroblastoma. And when you compare it to Crizotinib, if we look at the graphs on the left is Crizotinib, on the right is Lorlatinib. These are 4 neuroblastoma cell lines. The first one is wild type. The last 3 for both are mutated. And if you look at the IC50s, they're significantly lower for Lorlatinib when compared to Crizotinib. In addition, when you look at when Lorlatinib was studied in patient-derived xenografts of both Crizotinib resistant and Crizotinib sensitive neuroblastoma, you can see that Lorlatinib is far superior. So the black line are mice treated with vehicle control, the blue line is Crizotinib, and the red line is Lorlatinib. And you can see in all models, Lorlatinib inhibited tumor growth and improved event-free survival as compared to Crizotinib in every model studied. And looking further at why Lorlatinib may be more effective than Crizotinib, the phosphorylation of these mutations were studied when using Crizotinib versus Lorlatinib. And over here, this plot shows the activation loop of the kinase, whereas over here is the C-terminal regulatory tail of the kinase. And you can see when cell lines are exposed to Crizotinib and Lorlatinib, phosphorylation decreases in the activation loop of the kinase. But in the C-terminal regulatory tail of the kinase, Crizotinib really does nothing to inhibit phosphorylation, whereas Lorlatinib is quite effective. So this really may be a big reason why Lorlatinib is more effective than Crizotinib. Lorlatinib is FDA approved for non-small cell lung cancer, and the adult recommended phase 2 dose is 100 milligrams once daily. It's really quite well tolerated. Most of the toxicities seen are low grade, grade 1 to 2. And some of the more common toxicities, including peripheral neuropathy, cognitive effects, mood effects, can be reversed when lorlatinib is either dose reduced or held. So really, it has been shown to be quite well tolerated. So based on this data in adults, NANT, which is the New Approaches to Neuroblastoma Treatment, a consortium that studies, runs clinical trials in children with relapsed and refractory neuroblastoma, has been running a phase 1 trial of lorlatinib in children and adults with ALK-driven relapsed and refractory neuroblastoma. This trial is ongoing. It's being run by Kelly Goldsmith out of Emory, and it's studying both single agent crizotinib as well as crizotinib in combination with topotecan and cyclophosphamide. And I'm not going to read through all of these for the sake of time, but the goal is obviously to determine the maximum tolerated dose, safety, tolerability, and pharmacokinetics of lorlatinib alone and in combination. There are multiple exploratory aims of this trial, but one of the most interesting, I think, is that they're prospectively determining the overall frequency of circulating tumor cell-free DNA detection and the profile of acquired somatic mutations throughout study and treatment with lorlatinib. So they're enrolling children and adults with relapsed refractory neuroblastoma that's ALK aberrant. They are allowed to have received a prior ALK inhibitor, but not lorlatinib, and they're allowed to have CNS disease, given we know it crosses a blood-brain barrier. And this is just really to show the schema. They're treated with lorlatinib once daily, and then they go on if they don't have progression or a dose-limiting toxicity. But also to point out that the goal is to escalate to doses that are much higher than the adult recommended phase 2 dose. And the rationale for this is that really inhibiting full-length ALK in ALK-mutated neuroblastoma more closely represents what it takes to inhibit translocated ALK in non-small cell lung cancer that has acquired a resistance mutation. So on the top panel is what it takes to inhibit ALK in non-small cell lung cancer with translocated ALK without a resistance mutation. It requires only low doses of 1.5 to 3 mg per kg, whereas if it's acquired a resistance mutation, it requires higher doses of about 10 mg per kg of lorlatinib. And this is the same for our ALK-mutated cell line, in which you need about 10 mg per kg of lorlatinib to inhibit tumor growth. And so this extrapolation from this makes us think that it requires about 2 times the adult maximum tolerated dose to inhibit ALK in neuroblastoma. And this is just the schema for combining it with chemotherapy, which is really quite similar to what we did with crizotinib. So as we move forward, we're trying to figure out, make sure we're doing the best for our kids with ALK aberrant neuroblastoma. And the unprecedented single agent preclinical data of lorlatinib, as well as the established safety in both adults and children, which is ongoing, but we hope will be established shortly, gives us the impetus to actually amend our current phase 3 trial to replace crizotinib with lorlatinib. In addition, we're working with our European colleagues in SIOPIN and are working closely to develop identical trials that will run parallel so that we can sort of pool our data and hopefully work towards licensing for this indication. I think that the story of ALK and neuroblastoma, I'm going to rephrase that. We have ongoing and future challenges. One of our challenges is that we know that there's a high frequency of ALK mutations in relapsed neuroblastoma, with actually 40% of relapsed specimens harboring an ALK mutation. This is likely due to the presence of sub-clonal populations that are there at the time of diagnosis, but not detected by the most common ways in which we're using to detect these mutations, which is Sanger sequencing. And we know that deep and ultra deep sequencing techniques have revealed low frequency ALK hotspot mutations at the time of diagnosis. And it's likely that if we're not targeting these are going to be selected for and lead to disease recurrence in this patient population. So we really need to optimize our method for detecting mutations. And so along with our amendment to replace crizotinib with lorlatinib, we'll also be planning to switch our mutation detection method from Sanger sequencing to a next-gen sequencing panel. We're also interested in further studying the use of circulating tumor DNA and cell-free plasma for assignment to an ALK inhibitor. We know that a neuroblastoma is a very heterogeneous tumor, and that ALK mutations might be present in one portion of the tumor and not present in another. And a good way to make sure we're capturing this is through the use of circulating tumor DNA. And there have been studies that have been shown that mutations can be detected in the plasma of patients with neuroblastoma that are not detected in the tumor. In addition, we need to be worried about Lorlatinib resistance. We know this occurs through different mechanisms, and in neuroblastoma, it's thought to occur through EGFR and ERBB4, as well as, even more importantly, the MAP kinase pathway. So perhaps we need to be targeting MAP kinase pathway at the same time, but this is needs to be further investigated. In addition, this is just a slide to show that the quest for the best mechanism to target ALK continues. So people are looking at the use of antibodies to target ALK. And down here, you can see these are studies looking at antibodies in combination with Crizotinib, which actually in preclinical models show really nice results when you combine Crizotinib with a monoclonal antibody. But these antibodies are not really useful in the clinical setting. But what also has been very encouraging is the use of an antibody drug conjugate, which is this CDX015-TEI. CDX015 is the antibody, TEI is the drug conjugate, which is an alkylating agent. And you can see in this ALK mutated xenograft at the high dose of 10 mg per kg of the antibody drug conjugate, there's decreased tumor growth, increased survival. In addition, excitingly, there's a role for use of this antibody drug conjugate in an ALK wild type tumor model. And this is because we know that nearly all neuroblastomas express ALK, even if they're not ALK mutated. And so this is quite exciting, as this antibody drug conjugate might play a role in the treatment for all kids with neuroblastoma, not just ALK mutated. So I think targeting ALK in neuroblastoma highlights a lot of the challenges of pediatric drug development and the need to be nimble. There's a long timeline for development of these drugs. The metabolism in kids is different than adults. We need to do PK analysis in kids. We can't rely on the adult data. The genomic landscape of pediatric cancers is different from adult cancers, and the response to therapy differs. We also have to convince our institutions that we're working with and investigators to get large tumor specimens large enough for molecular analysis. And this is challenging in neuroblastoma because this has been a disease that historically a lot of people have just made off of the diagnosis off of bone marrow specimens alone. So it's a frame shift for a lot of investigators. And you can do ALK mutation analysis off of bone marrow. It's just more difficult. And then formulation issues is a real barrier for children as well. The FDA has a few mandated acts to support pediatric cancer drug development. In 2003, the PREA Act went into effect, but really was not helpful in accelerating drug development for children, because the FDA could waive studies in children if the disease for which the drug was being studied in adults does not exist in children. And orphan indications were exempt of pediatric studies as well. Hopefully, in August of 2020, so not too far away, when the RACE Act goes into effect, things will improve. And in the RACE Act, pediatric investigation is required if the drug or biologic product is intended for treatment of adult cancer is directed at a molecular target that the FDA determines to be substantially relevant to the growth and progression of pediatric cancer. So the FDA will publish a list of the molecular targets considered to fit this criteria, and this will trigger a new pediatric investigation requirement. And then the RACE will also amend the PREA exemption for orphan designated indications. So this is where we have been in the past, in which first in child studies, sorry, this isn't moving very well. First in child studies occurs several years after the first in human studies in adults, and even after FDA approval. And this is where we want to move to, where adolescents are enrolled on trials concurrently with adults. The pediatric trials, plans for pediatric trials occur much earlier than they do now, and even prior to FDA approval. And I think with moving this in this direction, we can improve the outcomes for children with horrible diseases like high-risk neuroblastoma. So I think we've come a long way, but we have miles to go before we sleep. And I want to thank several people, most importantly, Brenda Weigel, who is my mentor here at the U, but also through my mentor through the Children's Oncology Group, the COG Neuroblastoma Committee, and Pfizer, and really, most importantly, the patients and their families who allow us to take part in their care and take the risk of enrolling on our trials. So thank you. I'm happy to answer any questions. Sorry, I went a few minutes longer than I intended.
Thanks, Emily. So I might have missed this subtlety. So do you think that the frequency of ALK mutations had been, was incorrect, and it was thought to be 14%, and you guys were finding 7%, or is it related to the issue you brought up that specimens aren't always, you're missing mutations because you're not getting good specimens?
Yeah.
So what's the true frequency of ALK aberration?
Yeah, I've brought this up to our COG neuroblastoma group, and multiple studies have found the true frequency of ALK mutations to be 8 to 10%, and the frequency of amplification to be 2 to 4%. So I think that's true. We are definitely missing patients, and part of it is due to the sample submission, but it's not telling us the whole story. I happen to know of a few kids that unfortunately had tumors with ALK mutations that did not enroll on the trial. So I don't know if we're disproportionately not capturing that entire patient population as compared to the rest of the high-risk neuroblastoma population. There are definitely other factors involved, but I think the specimens do play a big role.
So, Emily, in some of the adult trials where we employ targeted therapy, one of the long-term goals is to de-escalate therapy as opposed to just add it to everything that exists.
Yeah.
So you pointed out the fact that there are a lot of late effects that are probably due to non-specific cytotoxics.
Yes.
So the pediatricians think that way? And I'm asking the question, if your trial looks like there's an induction and then some patients will go to surgery. Mm-hmm. In some adult cancers, and particularly in breast cancer, our observation is if the response is exceptional at surgery, meaning no evidence of invasive cancer, you may be able to stop there without the rest of everything. So do you guys think that way, or how are you approaching targeted therapies and thinking about de-escalation? Yeah,
I think that's a great question. And I think our ultimate goal in the pediatric oncology world is to de-escalate, replace some of the more toxic therapy with our less toxic targeted therapies, immunotherapies, et cetera. I think one of the, and there are definitely other disease groups outside of neuroblastoma, as well as some of the lower and intermediate risk neuroblastoma subsets of patients in which we are making those improvements. I think one of the challenges in high risk neuroblastoma is that how we've gotten to where we are today is due to a series of clinical trials through, previously Children's Cancer Group, Pediatric Oncology Group, now Children's Oncology Group, in which every time we've added something, whether it's a transplant instead of just continuation chemotherapy, or 2 transplants instead of 1, or immunotherapy, every time we've added something, we've improved the outcomes. And so in this specific disease, people are really hesitant to pull back, especially because our survival rates are still not where we want them to be. But it is a very huge topic of conversation within the neuroblastoma community. And I think one of the places we'll think about pulling back first, even though we just had the trial that showed that 2 autologous transplants are better than 1, I think that'll be the area that we focus on first.
Are there any more questions for Dr. Greengard? All right, let's thank our speaker one more time.
Thank you guys so much.